Good morning, everyone. It is 10 a.m. Eastern Standard Time, so we will get started with the beginning of our day. So just to briefly introduce myself for those who might not know, my name's Lauren. I'm currently the program manager at the 3Rs Collaborative. I'd like to welcome you all to day 2 of our workshop titled MPS Cell Sources and Suppliers. So just a quick disclaimer to get started. This webinar and workshop series is made up of companies who may be competitors or customers of one another. So accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So I'd first like to begin with an introduction to the 3Rs Collaborative. Then David will speak about the IQ and this webinar series in general. So the 3Rs Collaborative MPS initiative collaborates to accelerate adoption of MPS in scientific research. And our initiative seeks to increase adoption and regulatory acceptance of MPS in coordination with animal studies. And we do this through our diverse leadership. So this is just our board of directors on the screen here, but our members span across the scientific field. And as you can see, our MPS initiative itself has 42 institutions who are members, and we'd just like to thank all of them as we conduct this webinar today. So our initiative has 5 aims: to provide thought leadership and expert consensus from developers, to facilitate appropriate discussion, collaboration, and sharing, to create direct engagement between developers and regulatory agencies, to develop external partnerships and collaborations, and to create resources to facilitate increased engagement with commercial MPS developers. And we do this through 3 main work streams: our regulatory, our technology expo, and our end user engagement subgroup. So we're consistently engaging with end users and providing education. So just looking at 2024, we joined Altex as a member. We've conducted quarterly workshops, which this is the 3rd one, and we've had talks at the MPS World Summit, SOT, and Eurox. And you can look at our past webinar presentations from this year at our presentation page. And then our regulatory group has also had a lot of progress this year. Just looking at 2024, we have a publication submitted on perspectives on using MPS for regulatory applications, a PPP with FDA CEDAR, and a cross-platform DILI project. And then finally, our technology hub, which we launched in 2021, and we're continuing to add to this and improve this resource, and you can see our tech hub, the link there if you're interested in visiting. So I just want to remind everyone, as this is the 3rd webinar in our workshop series for 2024, the final one is coming up in November, which will be on oncology MPS, so make sure to save the date for that. And in terms of today's webinar, you will be able to submit questions using the Q&A box. Time permitting, we will answer these following each presenter, but if not, they'll be answered at a roundtable at the very end. All attendees will be muted with cameras off, so please reminder to use the Q&A box. I'll be monitoring the chat as well, but the Q&A is preferred. And again, you can visit our website to learn more and email me specifically if you're interested in joining our MPS initiative or becoming one of our sponsors.
So hi, everyone. My name is David, and I'm part of the IQ MPS affiliate. I specifically serve on our strategic partnerships and communications team. So as Lauren said, I just want to kind of give a brief introduction to the IQ MPS. So overall, we are currently composed of 27 member companies, And we have diverse representation from drug safety, 3Rs, ADME, and PD. And our current 2024 leadership team is made up of our chair, Anya Kopek, vice chair, Rhiannon David, vice chair-elect, Kim Homan, as well as our past chair, Rhiannon Hardwick. And besides the leadership team, we also are made up of a steering committee, and this is 2 individuals from every one of our member companies. And then to the right, just highlighting a little bit more about the IQ-MPS, we have over 150 active participants, and we have published several publications and working on several more that are soon to come. And also, I'd like to point people to our LinkedIn page that we recently created in the last year. So we post a lot about the IQ-MPS activities, so feel free to follow us there to keep up. The IQ is made up of 4 main goals. One being to serve as a thought leader for both MPS developers and stakeholder organizations. Another to provide a venue for appropriate cross-pharma collaboration and data sharing. Also to create focused engagement between industry and regulatory agencies. And finally, develop external partnerships and collaborations. We also are made up of 5 affiliate work streams. One being organotypic manuscripts team, and the goal of this is to publish manuscripts outlining organ-specific industry requirements for MPS platforms. Also, we have regulatory outreach, which identifies opportunities to engage with health authorities globally on topics related to MPS qualification. We also have a pilot project team, and this team initiates near-term proof of concept studies for data sharing or prospective collaboration for qualifying MPS systems. Also, the team that I'm a part of is the Strategic Partnerships and Communications team. And here we want to build relationships with key MPS stakeholders to enhance the inclusion of industry priorities. And then finally, we have a landscape gap survey team, which conducts and analyzes surveys, benchmarking how pharmaceutical companies are currently using MPS. And here I just have an overview of our external partnerships over the past few years, starting back in 2018. But we do have several other activities besides this group. And just to highlight quickly, in 2021, as Lauren mentioned, we co-partnered with them, which allowed us to work together on these webinars and workshops. And in 2022 and 2023, we've had 4 webinars each year on different MPS systems, such as liver, blood-brain barrier, vascular, GI, and neuro. And then this year, we also have, including this one, 2 previous ones and 1 upcoming one that Lauren mentioned in November that will be on oncology. In addition to our 3Rs collaboration, we also participated in several different activities with the FDA, such as an IQ-MPS FDA workshop, as well as upcoming webinars across different organ systems that we're working on with the FDA. So we're really excited to continue these efforts in the upcoming years. But if you have any specific questions on the IQ-MPS, feel free to reach out, or you could check out our website that I have in the lower left corner. But yeah, that's all I have today. I'm excited to get the webinar going.
Okay, thanks very much, Lauren there. So I'm going to just cover a couple of little things. I'm going to give you a reverse order. I'm just going to let you know a little bit about my consulting services and then a little bit about what I think the cell sources and suppliers are. There's a few things about me most of you know, but the important things here is I'm the 3Rs collaborative board member, and I'm also on the UK NC3Rs board. And I think another important one there is I'm the chairman of Matrix, which relates into a little bit of some of the things we've been talking about yesterday and probably today. I've got a wide variety of customers, from cosmetics, CROs, microfluidic organ chips, regulatory agencies, venture capital. So if you're interested, I'm more than happy to help you out. The consulting services are broad and ranged. So 3Rs considerations and strategy, in vitro toxicology across many different areas, but particularly in dermal and respiratory and organ chips. and animal-free solutions. I evaluate companies' products and services, and I'm a scientific expert witness. So again, there's a few things there. I evaluate competitive landscape, find technology providers, understand their technologies, which is brilliant because I'm a member of the MPS subgroup. Reports to the client, develop testing strategies, new test methods, project manage, writing papers and reports, support regulatory submissions, quality and process improvements. And I've also developed continuous improvement programs. All you little guys out there, you must have a continuous improvement program. Mentor and coaching. So I had a little bit, let's get to the most important part about why we're all here today. So I was thinking about what are the MPS supporting services, the sales sources and suppliers. And yesterday we heard some great big selection of different ideas about what they may be. So I certainly think they should be things like cell lines, tissues, organoid, or whatever you want to call your more complex test system. So I think they're those vendors, and some of those are a lot of those are our members, and some of them are MPS suppliers who just do that as part of their organ chip testing. Of course, it's equipment. That equipment can be pretty well simple lab equipment, general lab equipment. You know, we can't do things without good quality lab equipment, but it can be very specialized. I mean, I do a lot of work in the respiratory area, and we have lots of ways of developing in getting our inhaled substance onto our cells. So companies like that's useful, I can't remember. Anyway, so there's plenty of companies who generate systems to help us to do that. But it could be simple things like 96 well plates. There are many different things that can be suppliers to the MPS. Media suppliers is another one. Obviously, we're looking to try and move ourselves to using animal-free, chemically defined media. But how are we going to do that? And of course, and we're going to hear what the others are, where my mind isn't able to pick out those exciting things that you're all doing. So I just want to bring your attention to how can we create new test methods when there are so many animal-derived media, and examples include matrigel and fetal bovine serum albumin. As I said, I'm also part of the UK NC3Rs, and we've got a really nice piece here, moving to animal-free scaffolds for the construction of 3D organotypic models. Go there, it gives you lots of information, in vitro scaffolds, animal-free technologies, case studies, further reading and resources. But I'm going to make it easy for you because here are those resources. One of our speakers yesterday, Optala, they did mention that they had a product. They should look out here. If they go onto the website that I've got given here, you can click onto that, and it gives you the email that to contact NC3Rs about being included in here. But clearly there are many different products here, which means we don't have to be forced into using matrigel and fetal bovine serum albumin. We also recommend that if you're going to start developing, start with an animal-free. You've always got to develop your models and make changes to your media ingredients. So think about that when you're starting off. Don't just pick up on what other people have been doing. I represent PeptiMatrix, and we develop hydrogels to replace matrigel. We've got various different products. Core was our first product. We've got a product that is perfect for modeling tissues rich in fibronectin. We've got products for laminin, rich in laminin. And collagen mimics. And also we've got new things that we're working on. So our prime is under development. And we also make bespoke hydrogels for companies. So a little bit about that. If you want to know more, you can contact PeptiMatrix or just get in contact with me. I will also as I said, I'm a member of the 3Rs Collaborative Board. So I'm always going to talk about what we've got. Please use the website. There's loads of information on there. 3rc.org. If you go and put the S in there, you'll find yourself finding out about football. 3rc.org MPS presentations is a great place to look at our historical presentations. And I really can't say enough about the hub, the Microphysiological Systems hub. You can just click on the different boxes to find out the people who are doing different things. You can and then you can click onto the different companies and you go to their websites. They're all our members, so you're guaranteed that they're completely involved with the 3Rs Collaborative. I think that's enough from me. I'm looking forward to hearing all of the other presentations, and I will be helping in the panel discussion, chairing the panel discussion this later on today. And I think back to Lauren.
Okay, thank you for that introduction. I'm Gavin Knight. I am one of the co-founders of Neurosetta and the CTO. First, let me say, very grateful to be given the opportunity to present today. So as the title suggests, we're going to talk about the technology we developed called the Rosetta Array and how we use that as a quantitative high throughput screening tool for assessing human neurodevelopmental toxicity, which, despite the long name, is actually not very specific. So what we specifically do is generate models of human brain and spinal cord morphogenesis for in vitro risk assessment for this first stages of central nervous system development. So as you can see at the top of the screen, I've got a developmental timeline of central nervous system across the top, and I've got sort of the OECD's current guideline for developmental toxicity testing using in vitro assays. And so you can see there are 17 assays that really span this entire developmental timeline. So you have pretty good coverage, but you'll note that a number of these assays specialize in time points that are after the formation of the placental barrier and after the formation of the blood-brain barrier. And we know that, there we go. We know that there are a number of very important developmental stages that occur even in this first trimester, within these first few weeks, primarily initiation of the formation of the neural tube, which is the source of all central nervous system tissue. And we know that perturbations occur here that cause birth defects. And we're actually learning more and more that, slight perturbations that are fairly subtle can lead to autism spectrum disorder pathogenesis. So we've noted that, morphogenetic assays would be really useful here, and that pluripotent stem cell derived neural stem cell organoids can model a lot of the complexities of both forebrain and spinal cord throughout this time points, but that they're not been well used, at least since their inception, in large part because the tissues are very complex, and so they're really difficult to reproduce and thus scale. And so our assay, we think, sits in this early time point and allows us to really reproducibly model the morphogenesis of the forebrain and spinal cord to allow sort of supplement the current in vitro testing landscape with this potentially really useful technology. So I mentioned that we use human neural organoids, and so in order to understand sort of how our technology works at base, you got to talk a little bit about how the central nervous system develops. So it initiates within those first few weeks post-conception. Generally within the first 4 weeks, you get formation of the neural tube. I said that it is exactly what it sounds like. It is a tube of neural stem cells that spans the rostrocaudal axis, so the head to tail axis on that dorsal side, so the back. And if you were to cut the middle and then look down the barrel of the tube, upon initial formation, it's actually got a really simple single layer of neural stem cells that have this hallmark apical polarization of N-cadherin or tight junction proteins and this basal deposition of ECM. In this case, we're showing laminin. And what we and others have noted that over the years, where human pluripotent stem cells are differentiated in vitro towards a neural lineage, both in 2D and 3D, they initiate as these neural rosettes that have a very similar morphology, but they have the same drawbacks of typical organoids in that they form at random spots in the culture and at different shapes and sizes. So what our technology allows is to restrict where these pluripotent stem cells adhere when they're differentiating and adherent culture, and this allows us to have very tight control of both where they emerge and what size they are when they emerge. And we're able to do this really efficiently, greater than 99% in some cases. I mentioned that these initiate as 2D, but I'll say it's really important to note that they become multiple cell layers thick within the first few days of culture, and it's actually necessary for them to become sort of this pseudo 3D, hemispherical tissue to recapitulate this morphogenetic event. And you can see there, as we section through that, that's actually got a very conspicuous morphology in terms of like in vivo neural tube. But this ability to control that first stage of central nervous system morphogenesis in vitro allows us to have a really good starting point to begin to model that. And so that combined with our ability to generate neural progenitor cells that correspond to multiple different regions of the central nervous system, so brain and multiple different regions of the spinal cord, allow us to seed those cryopreserved progenitors directly on our custom plates. And in the course of 7 days, in the case of forebrain, and 5 days in the case of spinal cord, we can generate thousands of tissues per the usable area of a 96-well plate. How we do this for developmental neurotoxicity testing is generally 2 compounds per plate with 8-point dose response for each compound. And then for each of these wells, they may have between 220 and 1,000 single neurorosettes, depending on the size of the tissue that you're interested in, of which we'll sample 40 tissues per control. And then we will use our automated image analysis software, this Rosette Detect, that uses convolutional neural networks to assess the morphology of the tissue in the image. This allows us to get primarily 3 endpoints. So one, a measure of cell viability, another measure of neural differentiation efficiency, and then the sort of important metric that I've talked a lot about thus far, rosette morphogenesis or the rosette formation. This allows us to have a lot of statistical power, but also a certain mechanistic insight. Like we can see, where each specific endpoint is being potentially perturbed. And of course, because this is 96-well plate and we've automated quite a bit of the media handling steps, it's pretty scalable. So I'm not going to talk too much about sort of the detail of the results. I do want to talk about some bullet points that we've gone over to validate the technology thus far. We've confirmed that it works with compounds and DMSO solvents up to 0.1% with no effect on neural differentiation or rosette formation efficiency. We know that it has excellent reproducibility. So the forebrain rosette array across the first 33 assays that we ran were 86.3% plus or minus 9 single rosette emergence efficiency. So this means that every spot in a well plate that could have had a rosette, 86% or more of those were single neurosettes. The spinal cord, in this case the lumbosacrospinal cord rosette array, had even greater efficiency. That was 93.8% single neurosettes. And then as we began to run these sort of in a higher throughput fashion in 96-well plates with solvent controls and positive controls for that morphogenesis endpoint, we found that across the first 17 assays we ran, we had a Z-score of 0.54, which we felt was really robust. We have since over doubled the amount of assays we run, and we continue to add to this to sort of keep a running tally of the robustness of this assay. And so it's sitting at 0.532, which we find to be pretty pleasing for an in vitro pluripotent stem cell pseudo-organoid assay. We have integrated human metabolism, or a simulation of human metabolism, for this DNT screening. I'll talk a little bit about how that works. We use human S9 liver fraction to digest compounds, and then we will run side-by-side dose responses of the undigested compound and the digested compound. And we've seen really interesting results, particularly for compounds whose primary metabolites are known to be more cytotoxic or more neurotoxic, where we see an increase in sensitivity in those metrics in our assay. We've also found that you can get unique dose response profiles depending on which region of the central nervous system that you're assaying. We've done a number of compounds that cause spina bifida, which is a caudal neural tube defect, meaning it affects the lower regions of the spinal cord. We found that when we screen that compound with our forebrain rosette array, we don't see any effect. But when we screen that compound with our spinal rosette array, we see an inability of those spinal rosettes to contract and then to stop forming as we get to higher concentrations, which really sort of shows the utility of having multiple regions represented in the assay. And so that was a really cool finding. We have used our rosette array in-house to model 2 different genetic causes of spina bifida, which is that neural tube defect I mentioned before. There are 2 gene mutations among many that are associated with this, but the two principal ones we looked at were mutations in the SCRIB pathway, which would be associated with planar cell polarity, so the ability of cells to contract. And then the next one was folic acid pathway. The folic pathway is really well known, implicated in neural tube defects, and so we've modeled that both with our assay. Really interestingly, a collaborator of ours used the Rosetta array to model one of the genetic causes of autism spectrum disorder, which we found to be pretty surprising but very interesting. So I would encourage you to check out that publication if you're interested in that. Moving forward to sort of how we interpret things as they come off this assay, we sort of have a four-tiered assessment of risk. So if, for instance, there were no response across a compound for any of the metrics, say aspirin or saccharin in this case, this would be a no-risk compound. If you saw a response, but at, like say, exceptionally high concentrations exceeding 10 micromolar, we would say this is fairly low risk, considering that would either be a very high dose or a very high exposure. Where you enter into compounds that elicit IC50s between 10 micromolar and 100 nanomolar, we'd call that medium risk. So that would be sort of context specific. And as you get into the compounds that have known spina bifida causes, or just known to be sort of toxic at lower concentrations, this will be what we call high risk, where you're seeing responses in the picomolar or nanomolar response range. We utilize this, across the first few samples to then begin analyzing this at a greater breadth. We're lucky enough to work with the EPA. They sent us their ToxCast phase 1 and 2 libraries, and they were kind enough to help us prioritize these, meaning we could still keep it blinded, but we selected 117 compounds that had, the most screen compounds across all those 17 assays of the DNT IVB, so we could see how our assay kind of sort of complements that. And across these first 50 compounds, we saw a sensitivity of 87.1% and a specificity of 100%, granted very few negative controls. So that's not really that meaningful. We've since added, I think, between 20 and 30 more compounds from these plates onto here. So we don't have those unblinded yet. So hoping to get more negative compounds to sort of make this number more meaningful as we move on. That's the breadth of our work, is ensuring that this is a useful assay as far as like the classic compounds that have been run across the DNT IVB. I'll end here by saying sort of where we envision the Rosetta array platform being used in product development. So for drug development companies or agrochemical companies, given our ability to screen, we feel like would be very well utilized at this lead optimization phase, where we could sort of be faster than, say, animal models, and very competitive with other in vitro models, and sort of this with our unique utility of being a morphogenetic assay. So I'll say that our bread and butter right now is the contract human DNT screens, but we also sell the Rosetta array plates, and we sell credits for the Rosetta Detect image analysis. So if people are interested in seeing how the technology works, we can sell you a plate. And if you have neural organoids that are interested in being analyzed via our automated image analysis, and in fact, we're actually interested in using this for, say, non-neural organoids as well. So if you're curious about this, please contact us, because we sort of want to stress test this. As I said, we've pretty well established this for models of spina bifida and autism spectrum disorder. So we offer personalized disease model development, and we're currently building this out to be sort of a better precision medicine and drug discovery tool. But with that, I'll thank you for the opportunity to speak today, and I will gladly attempt to answer any questions that folks may have.
So I'll go ahead. So hi everyone. Thank you for tuning into today's webinar series, and thank you to the organizers, of course, for putting this together. So my name is Rachel. I'm a senior scientist in the Contract Research Services team. I will be presenting on behalf of CN Bio today to help you best prepare your organ-on-a-chip setup using the right cells and suppliers, whilst highlighting the importance of cell validation. So here's a very brief introduction about CN Bio. As you can see on this slide, we've come a long way since 2012 and contributed various technological advancements towards the MPS community, and that includes generating key data to support IND filing for liver disease drug. We are also backed by 14 patent families, and we have more than 50 peer-reviewed publications that demonstrates our technology robustness and also our the human relevance of our models. So this helped establish ourselves as a world leader in the microphysiological systems. And on this slide, I'd like to introduce you to our MPS platform known as the BiomimX. We have developed various multi-chip consumables plates for the liver, lung, and gut, with our most recent development being the high-throughput BiomimX model, known as the Liver 48. So our role at CM Bio is not just to develop the MPS system, but also to provide critical support to ensure that the success of our users, and that includes providing validated SOPs and national box solutions that contain our BiomimX 3D validated cells. So I'd like to introduce you to some of our BiomimX models. So we have three main BiomimX models at the moment, with the first being the liver model. And this liver model contains either 12 or our higher throughput model that contains 48 wells. It consists of individual wells that is subjected to microtissue perfusion. The liver cells are seeded into a scaffold to form a 3D donut-shaped structure. The next model is the barrier model, which is used with our gut or lung cells. The flow of fluid occurs in the basolateral compartment, and the cells are seeded onto the transwall, where it can either be exposed to liquid or have an air-liquid interface for lung studies. This model works particularly well for cells that require polarization, such as epithelial cells, and for transepithelial electrical resistance measurements. And our third model combines the liver and the barrier model and is known as the dual organ model. It allows the combination of multi-organ tissue study to study drug metabolism, absorption, or bioavailability. So across these three models, we are able to carry out various studies, particularly for disease modeling, such as NASH, hepatitis B, respiratory infection, or any safety toxicology studies, such as drug-induced liver injury, immune DILI, genotoxicity, and as I have mentioned, any of the ADME drug absorption, metabolism, or bioavailability studies. So I'm sure lots of you are probably quite familiar with the MPS system, and so I'm just going to briefly mention why it's important, particularly why we also carry out any animal relevant work. So mainly the MPS enhances preclinical studies by addressing two key areas, with the first being able to refine your preclinical experimental design. For example, in human studies, it allows for earlier de-risking of drug development. It provides a window of opportunity to make informed stop or go decisions, and the data can further reinforce decision-making for molecule selection prior to clinical settings. So in animal studies, the data allows for better understanding of species-specific responses, and it supports translatability of preclinical studies. For example, how the in vitro model compares to an animal model in vivo, and therefore the value of organ on a chip. Secondly, the MPS allows for an opportunity to be better prepared for the clinic. For example, it can be used to predict human outcomes for new drug modalities that lack suitable in vivo models. And it can also be used to generate translatable data to predict clinical observations and confidently progress to human trials. So this slide is to highlight why we focus on the use of human cells on our BiomimX, whilst also carry out studies on animal cells or any animal work as a regulatory requirement by the FDA. So following on from the previous slide, whilst we have worked with various cell types in the past, including iPSCs, our weapon of choice is primary human cells that are well characterized and isolated to high standards by our suppliers. So in this poster presented by the FDA at the Society of Toxicology conference in 2023, whereby primary human hepatocytes were used on our BiomimX to demonstrate variability observed between different donors and also highlights the importance of cell validation process. So in this poster, they shared that whilst pooled donors have shown success in 2D cultures, they have not shown similar outputs in 3D models. Pooled and average donor data can mask small variabilities as compared to selected and validated donors, which show reproducibility and reliability on our platform. So if, for example, in the figure on the left-hand side, the albumin production is significantly lower, basically less than 1 in pooled donors as compared to selected individual donors on the right figure. It is also important to note that each donor also demonstrates variability in albumin production, but this is variability and responsiveness that can be properly characterized when the cells are validated. We'd also highly encourage people to take advantage of donor heterogeneity, as this reflects the actual human population and can be used to explore donor variability. This is where a high throughput system such as our BiomimX Liver 48 can be useful for testing of multiple donors across one plate. So our in-house contract research services team at CN Bio routinely validates and qualifies our cell sources on our platform prior to distributing to clients as part of our kit. So in this slide, I'm going to share with you an example of our stepwise cell validation process for non-alcoholic steatohepatitis, which uses a triple culture of cells, namely the hepatocytes, the Kupfers, as well as the stellates. So first of all, we of course have to ensure that we use multiple vendors to maintain a continuous cell supply, particularly after going through so much time validating those cells. Next, we carry out our validation on the primary human hepatocytes first. And we do so by running various cell health assays such as the albumin and urea production. We also check for CYP3A4 and LDH. To ensure reproducibility, we run 2 separate experiments with replicates. Next, once we've selected the hepatocytes that we have validated, we then move on to test the hepatocytes with our Kupfers. We then carried out another 14-day cell culture experiment, whereby we then proceed with the same cell health assessments, but also include a test with lipopolysaccharide to assess responsiveness to inflammation. Lastly, we perform a full NASH experiment containing the triple culture of the validated hepatocytes and Kupfers, and including the stellates. Again, running them with the same cell health assessments, but testing them with the absence or presence of TGF-beta to assess fibrotic response. So it's important that we run the validation QCs, including any replicates at each stage of this validation process to build confidence in our cell sources. So in the figure on the right, it's an example of the variability of albumin production that can be observed between different donors of primary human hepatocytes, whereby a poor quality donor produces significant lower amounts of albumin as compared to a high quality donor. So here's an example of our 14-day NASH experiment. So prior to day 0, we carry out any equipment QC, followed by the priming of our consumable plates. On day 0 itself, we carry out the seeding of our selected and validated cells. This is followed by incubation and media exchange. From day 4 onwards, we carry out our QC cell health assessments, and that includes confirming the cell functionality as well to test for albumin, urea production. If any dosing or treatment is also planned as part of the study, the QC steps are ran on each day of dosing to understand the effects of the treatment on cells and how the cells may behave in response to inflammation or any drug metabolism. The standard timeline for our cell validation process is up to 14 days with options to prolong the culture. And at the end of the experiment, various endpoint analysis can be carried out, including bright field microscopy, flow cytometry, and the scaffolds can also be immunostained and imaged with confocal for further analysis, and the cells can also be lysed for PCR and sequencing. So from our validation process, we've learned that more than 60% of primary human hepatocyte may fail to form 3D tissue in our model. The validation process is both cost and time consuming, but the benefits outweigh the cons, as we are then able to demonstrate greater chance of success on our platform in terms of reproducibility and reliability, as well as maintaining functionality of the cells for longer term cultures. And due to the quality checks that have been surpassed, we can also be confident about the data that is being benchmarked against. Lastly, cell validation allows us to pick characteristics such as known genotype and phenotypes for specific uses or to meet client needs. So here I'd like to share with you some of the suppliers that we are currently working with for our supply of primary human cells. At CM Bio, our focus is on the use of primary human cells because they are known for their excellent phenotypic relevance. It allows for exploration of donor variability and also the study of clinically relevant biomarkers. And it allows for longer cell cultures on our platform. So we partner with large cell suppliers who provide cells that have been isolated to high standards and are well characterized. Our main suppliers of primary human liver cells are LifeNet Health and Lonza, with Lonza also supplying us with primary lung cells. We also have a collaboration with Altis Biosystems for their replica kits, which contain primary human jejunum cells that we use in our bioavailability studies. We have worked with Lonza, and LifeNet Health to set up these agreements so that we can continue providing these validated cells to our customers. And this allows our models to be more accessible, as customers don't need to worry about whether the cells will work, as we've already validated them on our platform. So I hope that this has been helpful to the audience if they were planning to run their own in-house cell validation. Thank you.
Thanks so much, Rachel. That was fantastic. And I know I definitely learned a few things. It looks like you have a question in the Q&A, so I'll just read it out to you.
Okay.
Do you provide brain organoids, and where can we purchase such organoids?
So we do not provide any brain organoids as far as I know of, and we're definitely not in the process of validating any at the moment. It is something that we may explore, and particularly the assay development team may explore some the brain in future developments. We do not supply them, but it may be worth reaching out to any of the suppliers that we work with in case they do have any of that.
Great. Thank you, Rachel.
Thank you.
And good morning or good afternoon, depends where you are, to everybody. My name is Luana Ferrara, and I'm the head of R&D and collaboration here at Qkine. And today I'm going to give you a short presentation about the impact of high-quality recombinant growth factors on advancing animal-free research. So we all know that animal-free growth factors are crucial for researchers that want to create a more chemical-defined and controlled cell culture system, especially in those fields like tissue engineering and cell therapy, where consistency and regulatory compliance is a must. As you all know, the FDA no longer requires animal testing for new drugs and has been actively encouraging alternative methods to animal testing. Organ-on-a-chip is a very new promising alternative that aligns with the FDA screen for drugs or disease modeling or tissue engineering. As every other technique, however, organochip or organoids can have a few limitations or challenges. Those can be variability, problem in reproducibility, in standardization or scalability, and also come with a cost and high technical expertise. So how Qkine does fit in this environment, or how can we help scientists to progress in this field and address those challenges? Qkine is a specialist in manufacture of growth factor, animal-free growth factor and cytokine. The company was founded in 2016 in Cambridge. It's a spin-off for a laboratory from the Department of Biochemistry at the University of Cambridge. And we have a unique recombinant protein manufacture technology, and we have a range of improved growth factors. They're mostly human, but we also have specific range of species-specific growth factors for emerging fields such as organoid culture or cellular agriculture. How do we produce our animal-free growth factors? So we use a microbial expression system, particularly E. coli, and it's produced in our animal-free manufactured seed. Everything that comes into contact with our product from the very beginning of the chain till the very end, when the product is purified and tested, has to have an animal-free certificate. That means that every plasticware, chemical, enzyme, broth, compound, column, you name it, has to have a certificate of an animal-free before it enters the lab. This will ensure that at any point of the chain, or manufacture chain, that the product has been touched or contaminated with any animal product. The gene is usually the gene of interest that expressed for the growth factor interest is transformed into the bacteria cells, and then we induce the overexpression of the growth factor within the bacteria. The bacteria is going to use broth, so that's of course animal free, and the growth factors that is inside the E. coli cells is extracted. They usually express as an inclusion body. This due to the fact that the growth factors, human growth factors, extremely complex molecules, so they are expressed as an insoluble inclusion body. So we need to extract and solubilize the inclusion body and then refold those in vitro. And the protein is then purified at the maximum level of purity and is controlled is checked for make sure that it is achieved the correct conformation. We use a different array of biochemical methodology to check that the conformation is the correct one, and for example mass spectrometry or STS gel. And every batch of protein is produced is checked against mycoplasma. So with mycoplasma tested, we make sure they are sterile, and we test against a different range of anaerobic and aerobic bacteria, and is of course bioactive, and is always the same activity from batch to batch. We also test for endotoxin level. I'm going to give you now a couple of examples of how the animal-free growth factors compare to mammalian express or conditioned media growth factors. The first example is TGF-beta1. So we have produced the first completely animal-free TGF-beta1, and TGF-beta1 is a member of a wider family of transforming growth factors. Is the production of the complete animal-free TGF-beta1 allowed direct comparison with quality matched TGF-beta2 and 3 for serum free testing. In the left side of, let's see if I can show. In the left side of the slides, we show the activity of the animal-free and microbial expressed TGF-beta1 in green, and the two different suppliers, the supply mammalian express in black. As you can see, this we run an assay now, a cell-based assay, and we measure the binding and the activity of TGF-beta1 to the receptor. As you can see, the microbial animal-free TGF-beta1 is more effective than the mammalian express TGF-beta1. As you also can notice, that we tested 2 different batches of our TGF-beta1, and the activity is pretty much the same, showing how consistent from batch to batch. On the right, you can see the use of the TGF-beta1 and FGF2 for maintaining pluripotency in MPS culture. On the right panel is the microbial expressed Qkine TGF-beta1 and FGF2. On the left is the mammalian expressed. As you can see, the expression NANOC, there is a pluripotency marker, is more visible and more homogeneous when you use microbial expressed animal-free growth factor. This is due to the fact that the samples, so the growth factor is much more homogeneous from when it's produced from a microbial environment. The other example that I want to give is respondin-1. Respondin-1 is a growth factor that you use to potentiate the Wnt signaling in many cultures, including organoids. The majority of scientists still use respondin-1 from conditioned media. When you use conditioned media to your culture, you don't just add that growth factor that you intend to add, but you add all a series of compound and contaminant that comes from the conditioned media. So you're adding variability to your experiment. Not only that, any conditioned media is different from the previous batch, because whatever is inside the cells that are in the conditioned media is not controllable. It's not a defined system that you can actually control. So it adds a lot of variability. We have collaborated with Dr. Dennis Blanker from the Cold Spring Harbor Laboratory to test our respondin-1 in pancreatic organoid. Again, it's now conditioned media respondin-1. And on the right side, you also have a negative control. And as you can see, the Qkine recombinant respondin-1 is able to support the pancreatic organoid growth. And as a plus side to that, you know that by using a chemical defined cytokine, and you have more consistent results. You don't have to depend on the variability of the conditioned media growth factor that you add to your cells. Qkine does commit to 9 quality points. I'm not going to go through them all, but the most important that we are certified animal-free. So as I said before, there's a full traceability of all the components that get in touch with our product that are, of course, animal-free. We operate in an ISO 9000 manufacturing facility. We do rigorously test for accurate recovery, and I'm going to say something more in the next slides. The activity from lot to lot is consistent. That means every time you buy from us, you know that you don't have to buy from the same lot just to be sure that the activity is going to be the same, because it's the same from lot to lot. As I said before, we test for sterility and mycoplasma, but we also test for endotoxin level, and ours have the leading lowest level on the toxin. It's very important, and when you buy for growth factor, you actually check the endotoxin level of the growth factors, because this can have effect on your cells. And particular cell lines are very reactive to endotoxin and can be extremely disturbed from endotoxin. So it's important that this a parameter that should be looked after when buying a growth factors. This is a particular nice slide. I really like it because it shows how much care we put in our processes. If you work with growth factor before, I'm sure a lot of you have, you know that they usually arrived in lyophilized form, so you have to add a buffer to resuspend the growth factors and recover the protein, and then you dilute your you create your working dilution, and then you start your experiment. So when you add your buffer to resuspend your growth factor, you expect to recover 100% of the growth factor in the vial. So if you bought 100 microgram of whatever growth factor, you expect to recover 100 microgram of that. So by doing different comparison studies, we have seen that although we maintain a very close percentage of recovery, close to 100%. A lot of other suppliers don't. That means that these are another level of variability to your experiment, because sometimes you can recover even just 25%, sometimes 50%, sometimes even under 10% or under 20%. So it's very variable, and this variability has an effect on your experimentation, an effect on your cells. This particularly important when you work with growth factors that act as amorphogens. So their effect different if it's used in one concentration or another. So it's particularly important to know what a concentration of growth factor you're actually starting from. So this is a very nice graph that we always show to underline how accuracy is important for us. To just summarize how Qkine can help address those challenges. So we supply consistent high quality growth factors. Those growth factors will help to enhance reproducibility and scalability. This because every lot is compared to the previous one, and we make sure that nothing has changed. So you don't have to, so you're sure that whatever lot you're using is the same as the one they used before. The final culture system, how do we help that? So you can use our animal-free growth factor to create a more defined and controlled system for your experiment, and trying to align as much as possible to the FDA guidelines. Tailored growth factor. So we do work closely with academics and biotechs to offer support to their challenges, and we have a specialized group here of bioscience scientists that are always very happy to help our customer and future customer with their question they have regarding the cells, organoids or organoid chip. And we do offer cost-effective option as well. That's the end of my presentation. If you have any question, please feel free to ask. And I left my email address there, so if later on you want to contact me, please feel free.
Thank you. I'm Erika Ferrari. I'm commercial science leader in BiomimX, and thanks for organizing this very interesting workshop. And just a couple of words about BiomimX. So we are a spin-off of Politecnico di Milano, and the company was founded in 2017. We are 11 team members with 3 main departments in the company, so focused on commercial, R&D, and industrialization. We have a high track record of EU grants and many publications in the field, and our models are providing data for IND applications. What we mainly do is to partner with the pharma and the biotech by providing innovative tools to fostering their drug development pipeline. And let's say the field that we are working with is really relying on dynamic movement in our body. So as you may know, our organ in the body are subject to motion. Of course, the heart is the key example because it's beating, but there are many other organs that are subject to motion, such as the gut that has a peristalsis, the lung are breathing, and also the cells in our body are placed in a 3D microenvironment. So what we do in BiomimX is to really couple these 3 key features. So by using human cells in a dynamic and 3D environment to generate innovative preclinical models. As cells, we usually use human cells, cell lines, primary cells, IPS, patient-derived cells. We embed them in an extracellular matrix that can be different. So we use fibrin, collagen-based hydrogels, also PEG. And once the cells are embedded in this matrix, we put them in our platform, which is an organs-on-chip device. Here in the image, you see 3 main chambers in one device. And then we apply our peculiar mechanical stimulation to generate physiopathologic tissues. And the key advantage of the mechanical stimulation is that we can use it to both differentiate and maturate human tissue in vitro, but also we can tailor it to really mimic complex disease models. And this is the one chamber of the device. And from the top view, you see it being a, let's say, standard microfluidic device. And from the cross section, imagine here you have the cells in blue in 3D, and this membrane is moved so that this movement is transferred to the cells so that they will feel the movement itself. And in this video are cardiac fibroblasts that are being stretched, but by really, let's say, tailoring the geometrical parameters of the device, we can provide a different kind of mechanical stimulation. So we can provide stretching, so the cells will feel an elongation in a predefined direction, as you see here in the video. But we can also provide compression, so the cells will feel constrained within the channel itself. And by taking advantage of the channel geometry of the device, we can also develop barrier-like models or culture multiple cell types in an adjacent way. And this technology has been applied in different contexts of use. So BiomimX started to mainly work in the cardiac space, both physiologic and pathologic, but also on joint-related application, and in particular on osteoarthritis. And more recently, we also started to work on barriers such as the gut and the lung. And today, I just wanted to introduce these models and the way we characterize them. So starting from the cardiac model, our cardiac model is called UART and is made of 75% of IPS-derived cardiomyocyte and 25% of dermal or cardiac fibroblast, and they are embedded in a fibrin hydrogel at high concentration. And once they are injected in our model, we provide the mechanical stimulation to train the cells so that after 1 week of culture, they really appear interconnected. Here in the immunofluorescence staining, you can see the characteristic striation of the sarcomere, or you see in this video that the cells really beats all together as a syncytium after 7 days of mechanical stimulation. And the tissue is also functional because we have a mean of precisely insert electrodes within the device, so we can also measure characteristic electrophysiological parameters. We can also do gene expression, and in particular, we saw that the mechanical stimulation allows an increased expression of ion channel related genes, and in particular the potassium genes, and we can also do calcium transient analysis. And the advantages here of using IPS-derived cardiomyocyte for us is that they have the ability to recover functionality and their characteristic beating, which is very important for us so that we can acquire a clear signal readout. The challenges we mainly face is that as they are commercially available, there are different protocols with which they are developed. They are usually provided as single cells, so they would need initial manipulation before insertion into the device. And we also work in the cardiac space on genetic diseases, and these projects are done in collaboration with the hospitals in Europe, and we are mainly working on dilated cardiomyopathy and laminopathies. And what we do, we take patient-derived cells, we put them in our device, and we subject them to mechanical stimulation, and we see that within the device, these cells preserve patients' genetic background and recapitulate specific disease-linked phenotypes. And with this model, as well as with the standard UHeart model, we can test pharmacological responses and also contractility parameters. And here, the advantages of using patient-derived cells for us is that we can study different genetic mutation, so also helping us modeling the variability across individuals. The challenges we mainly face is that there are lab-to-lab differences in differentiation protocols of these cells, so we need to rely on lab optimization and characterization, and also that these cells must be easy of use to be incorporated within the platform. Concerning the cardiac fibrosis model, here what we do, we start by using healthy cardiac fibroblast. We embed them in a fibrin hydrogel and we put them within the device. And in this case, we use the mechanical stimulation to really trigger the pathology onset. And this is very important because usually cardiac fibrosis or fibrosis in general is developed through a cocktail based on TGF-beta, which can have a confounding effect when there is the need of testing the efficacy of drugs. So we are able to provide the same fibrotic traits with only the need of applying mechanical stimulation. And here you can see that if we compare the condition where we have only dynamic condition compared to the static condition with the TGF-beta, we see that we can have even higher phenotype switch from fibroblast into myofibroblast, as well as matrix deposition, and I'm here reporting the example of fibronectin. So here the advantages of using primary cells are that they have a closer representation of in vivo condition, but often there is a variability between batches, and they have a limited lifespan, so sometimes it's difficult to capture them for long-term studies. In the context of joint-related application, and in particular on osteoarthritis, here again, similarly to cardiac fibrosis, we take advantage of the mechanical stimulation to induce the pathology onset. So again, here we start from healthy primary chondrocyte. We embed them in hydrogel, we put them in our device, and we culture them for 2 weeks of static condition so that we can achieve a mature and healthy cartilage on chip model. And after these 2 weeks, then we start by inducing the pathology by means of hyperphysiological mechanical compression. And here we see that we see an increase of genes related to degradation and inflammation, and also the position of matrix. And once the model is developed, we can use it to test the efficacy of compounds. And here I'm just reporting the example of the comparison between the uni model and 2D multi-well plate. And by comparing these, when we test compounds, we see that only the uni model is able to respond to this compound. So meaning that really recapitulating the mechanical environment is key for osteoarthritis related application. And here the advantages of using primary chondrocytes is that, of course, they have a closer representation of in vivo condition, and also they in 3D and under mechanical stimulation, they are able to maintain their chondrogenic potential, which is often lost in 2D standard Petri dishes. Concerning the gut, UGUT is our model of intestinal barrier. And here from the video, you see that we apply a mechanical stimulation that really mimics the peristalsis. And we started by developing the models by means of cell lines, and in particular, we used the CACO2 cells and HT29 cells for the epithelial barrier, and HMEC1 cells for the endothelial barrier. And if we saw that if we provide a peristaltic-like movement, the cells in the epithelial compartment really generate villi-like structure with the increase of proliferation, maturation, and mucus production. And also the barriers after 7 days of culture appear tight. So here we flow two fluorescent dyes, red in the epithelium and green in the endothelium, and we see that the two dyes remains confined. And we are now using this model for absorption studies and microbiota analysis studies. And the advantages of using cell lines is their consistency and reproducibility and the standardization, and they are cost-effective and readily accessible, but they often lack the physiological complexity and genetic diversity of primary cells. So this is why we are currently further validating the model with the IPS-derived intestinal organoids. Concerning our liver application, so we started by developing a 2D liver model based on the micropattern co-culture model of hepatocytes and fibroblasts based on the development of Professor Qkine and Bhatia. And in particular, we started by means of cell lines such as FG2 and fibroblast, and we used this 2D model to be coupled in a multi-organ on-chip device of liver and heart that was used to study of target cardiotoxicity of liver metabolized drug. And then we started to work on a 3D liver model where instead we moved to primary human hepatocytes, so primary human cells for the hepatocytes, while sticking to cell lines for endothelial cells. In this case, we are using UVEC. And what we are doing in this model is to really try to recapitulate the characteristic of the liver sinusoid, so also trying to mimic the space of disc that is found between hepatocytes and endothelial cells. And we saw that this configuration allowed a higher functionality compared to the 2D model or to a similar model where the space of disc was not recapitulated. And here, the advantage of the cell lines are, again, their standardization and their cost effectiveness, but the primary human hepatocytes gave better results as they have closer representation of in vivo condition and higher functionality. However, they have a limited batch availability, and also the UVEC that we are using are not the best cell type for liver. That's why we are now moving towards LSEX, but they are also difficult to source. So some challenges here for what we are doing on the liver model. So just to conclude, BiomimX provides a cutting-edge, reliable cell culture platform for seamless integration into the drug development pipeline. We have strong expertise to generate relevant in vitro human models by means of using diverse cell types, including cell lines, primary cells, IPS, and patient-derived cells, to generate human-centric models, while also have an eye on the challenges of selecting the most suitable cell type for accurate results. And thank you for your attention. Here are our contacts if you wanted to know more about our models.
Thank you for the introduction and so much also for the invitation. And thank you everyone for joining. So my name is Linda and I would like to discuss our idea and how we use different cells to set up our models. So as a start, I represent Mimetas. So we are an organ-on-a-chip company where our headquarters is based in the Netherlands. And we develop human tissue models that would serve the development of therapeutics for this 21st century. So I'm scientific project lead in the model development team, working at the company for almost 8 years. And I'm also almost finished with my PhD, which focusing on disease modeling and specifically on kidney biology. So today I would like to discuss how we would use different cell sources to set up our model and how we would approach our approach on using these different cell sources. And I would like to show that by discussing a case study on renal inflammation. So as you may all know, cell sourcing is very important in order to support the drug discovery pipeline. And how we would envision that is that you basically have different puzzle pieces of setting up your assay. So the first part, of course, is setting up your model with a reliable source of biology. So at Mimetas, we work at different organs, different tissues, for which you can see a few examples here, including the kidney, the liver, and the gut. Next to that, the next puzzle piece is how we would induce a disease. So for that, you can use different cells with a specific engineered defect, for instance. But you can also think of an external trigger to add the trigger and induce the disease. And the final puzzle piece will be the readout to confirm the right mechanism of action, the right phenotype, or the way it should behave. And we can tailor the different puzzle pieces in order to set up the assay. And with that, we can serve different purposes, which are aligned on the bottom of the slide. So either you can use the assay to understand the biology. So a lot of diseases are very complex. So you can tailor your biology based on adding more cells, for instance, different disease triggers to understand the disease better. The next thing, once you have the disease biology, you can also, it could lead to the discovery of novel targets and drug candidates. You could also use the assay to do compound screens and prioritize which compounds to continue with later on in the pipeline. And of course, it can serve to de-risk and accelerate the development. So if we look at the approach that we use at Mimetas to select the cell source for setting up our models, roughly you would have 3 different cell sources. You can use cell lines, stem cell derived material like organoid, iPSC, or adult stem cell derived, or you can use primary materials. And we basically have a fit for purpose approach. So based on different characteristics, we make a selection. This can, of course, be the project goal. The characteristics of the cells, like the phenotype or expression of a specific receptor, for instance. Availability of cells, very important. Our previous experience with the cells, and of course the cost. So we work together with customers, for instance. We need to make sure that these cells are available commercially in order to perform the work. And then I move on to one of the cases that I would like to discuss. So that's focusing on renal inflammation. So our microfluidic platform is called the organoplate. We have different flavors of the organoplate. The one that we use for this project is the organoplate 3-lane. It has 40 chips on one plate. And what you can see in panel C is how the in vivo proximal tubule would look like of the kidney. So on top you have your proximal tubule epithelial cells. In the middle, in blue, you have an interstitial space of extracellular matrix and proteins. And on the bottom, you have your endothelial vessel with immune cells in the lumen. And in panel D, you see how we would, a schematic of how we would set it up in our organoplate chip. So in the organoplate 3-lane, we have 3 channels in one chip. The middle channel is used to deposit extracellular matrix. In this case, we use collagen 1. Then on top, we seed proximal tubule epithelial cells. And in the bottom, we seed endothelial cells. Upon perfusion, these cells start to form tubular structures against the extracellular matrix so that they are in direct contact with the ECM. And then finally, we add primary monocytes into the lumen of the endothelium, and we actually label them with the fluorescent dye so we can track them over time. So in this case, we set up this model as a healthy model first and characterize it. And then we wanted to study the interplay between the renal cells and the immune cells, which is very important in renal inflammation, not only for the development, but also for the progression. And we use this model to test immune modulatory compounds in the context of renal inflammation. So this work was done in collaboration with Sanofi. And we tested a bunch of compounds which they used to prioritize which to continue with. So as I mentioned, we have 3 different cell types, the RPTEX, endothelium, and immune cells. So the first step is characterizing the model as it is. So we first looked at the epithelial compartment. So for that part, for the RPTEX, we used the cell line. And what you can see here in this panel is a 3D reconstruction. So on the right-hand side, we have the RPTEX tubule, and on the left-hand side, you see the endothelial vessel. And in the middle, well, you don't see it, but there's the extracellular matrix. And what you can see is that both of these tubules have a clear lumen, as expected, and that's one of the things that we would like to have. And we did immunofluorescent staining to characterize polarization of the cells, as well as we looked at barrier formation. So you see a tight junction marker with CO1. We did the same thing for the endothelial compartment, and here we actually use primary cells. So we use primary UVAC from pooled donors. Also here we used immunofluorescent staining to characterize the cells, and you can see that for the tight junction and adherent junction markers, we have fecal adherent CD31, nicely expressed. And also here we also see polarization with the acetylated tubulin, representing primary cilia, and CO1 for tight junctions. So indicating that even though they're different cell sources, they have the intended use and phenotype as we would like them for this project. So once we have characterized the model, we added primary immune cells to the lumen of the endothelium. And to induce inflammation, we use complement activated serum, which is added to the renal compartment. So that's the compartment on top in yellow, where you have the proximal tubal epithelial cells. The complement activated serum starts to form a gradient towards the UVAC compartments, here in red called donor compartment. And upon this gradient, this complement activated serum is recruiting these monocytes. So you actually can see that these are moving out of the endothelial vessel into the extracellular compartment, into the renal compartment. As you can see in the video on the bottom, because we fluorescently labeled them, we can track them over time. What you can also see in the middle panel is that the trigger is inducing morphological changes in the RPTEX. So you see clustering of these cells, indicating tubular stress in the epithelial cells. Upon further characterization, we can see with immunofluorescent staining that the trigger, the Cas trigger, is inducing changes in the expression of acetylated tubulin. And this regulated tubulin is actually known to be a sign of tubular cell dysfunction. So you can see that with Cas, there's a different expression, as well as the expression of ICAM1. So we also saw ICAM1 expression in the UVACs coming up, but more interestingly, we also saw higher expression in the RPTEX cells. And of course, ICAM1 is an adhesion molecule that would help with the adhesion of immune cells and the recruitment. And what we did as well is, so we used primary immune cells from different donors. So we characterized different donors, a total 4. And we evaluated their migratory behavior over time. So here on the bottom in the graph, you can see the movement of these monocytes in different compartments, specifically in the renal compartment, after 96 hours. And you can see that with the trigger, so with the Cas trigger, all of these donors respond to it as there's more movement into the renal compartment. But you can see that there's differences, especially with donor 4. So that's something that we can pick up with this assay as well. And finally, we have been using this assay to evaluate immune modulatory compounds from Sanofi. So we have compound A and compound B. So the left-hand panel shows compound A, which is triggering the Cas trigger. And on the right-hand panel, we have compound B, which is targeting the monocyte specifically. So we can show that we have different compounds with different targets. And on the left-hand panel, you can see compound A. So there we use different strategies. So we either use compound A in the renal compartment, in the UVAC compartment, or in both compartments. And what you can see is that upon the trigger, you can see very nicely that there's an increase in the number of immune cells in the renal compartment, but that is blocked or inhibited as soon as you have compound A there. And on the right-hand panel, we see more or less the same thing with compound B, although here we tested different concentrations. But you see that we can significantly inhibit the movement of these monocytes in this assay. And that serves as a proof of concept for studying renal inflammation, but also for drug testing. So overall, this was just an example of showing that here we selected both a cell line as well as primary cells, which were not donor matched. But that can actually be sufficient to show that we have the intended phenotype for the purpose of the project that we have. And we've set up an immunocompetent in vitro model of the proximal tubule. And we showed that we could successfully show testing of immune modulatory compounds as a proof of concept. And with that, that's actually the final slide that I have. So if you're interested in any more of the research we're doing, we're not only working on the kidney, we're working on a lot of different tissues and organs. We also have different ways to work with us. You can work with the organoplate yourself, but you can also work together with us. So if you're interested, feel free to reach out or visit our website to have a look. So thank you so much for your attention, and happy to take any questions.
Thank you so much, Linda. That was really interesting, and taking us through the case study was really, really interesting as well. I'm not seeing any questions yet. Oh, I think one just came in, so I can read that to you. It says, thanks for the nice presentation. You introduce inflammatory cells, and then in brackets with a question mark, autologous. And if not autologous, the system can generate inflammatory response without adding the CAS. Could you please elaborate on this? And then it says also, how are you perfusing the cells? Does the whole blood running through the system or median and shear stress? Please elaborate. Just let me know if you want me to repeat any of that question.
Yeah. Let me start with the first question. So that's the type of immune cells that we use. So indeed, they're not donor matched. And of course, we always have our control without CAS. And we see that there is some baseline movement of these monocytes out of the endothelial vessel. So that could be a sign of that there is some baseline inflammation ongoing. But the most important thing here is that we have a decent assay window. So that's something that we have established between the non-trigger and the triggered condition. And since this assay window is big enough, we are able to accurately determine an effect of a compound. The second question is on how we perfuse the monocytes. So these monocytes are indeed in medium. So there's perfusion through this lumen of both of these tubules, which is just medium, and we can just add the monocytes through the medium. Perfusion is done by putting the plate on a rocker platform, so there's no pumping or anything going on. It's just bidirectional flow through gravity. We put the plate on top of a rocker platform, which is in the incubator, and with that we can just set the angle and you create flow, and with that the cells experience shear stress. I hope that answers the question.
Great. Thanks, Linda.
Great. So thank you again for letting me the opportunity to introduce what we do and what is the relationship with our cell providers. I'm going to give first a global overview of the technology we're providing and some use cases as the previous Mimetas very nice presentation. So we are a French-based company. Sorry about that. We're a French-based company, and basically our mojo is to reuse the fact that neurons innovate everything into your body, and that we can use those neurons as a natural biosensor that translates an inference of a compound, a chemical compound, or a new drug that is tested on the targeted organs. And the idea is to use the neurons as a sensor to digitize human biology. So how does it work? As you know, neurons has at the endings some very specific markers, receptors that are unique in each organ. For example, in the skin, in the liver, in the gut, you have all neurons from different that all are connected from peripheral nervous system, and at the endings and when they are connected into the epithelium, they have very specific receptors. And the idea is to take the advantage of the integration of a signal when there's something that's interacting with the receptors, and the transducing of the signal into a digital signal. Basically, when you have a baseline of whatever receptors that you have, we record this baseline, and when we apply a compound, we have a very specific response, again, electrical activity response that is going back from the endings to the cell body that are in your spine. And what we have observed is that each compound have a very specific electrical response, and that is encoded in a very specific way. So we take profit of this unique capability of neurons and different type of neurons to encode the effect of a compound. And basically, we have a lot of different metrics that characterize this signal, and up to 42 I'm going to show you after, and what we called a digital signature of a compound of a condition. And the global idea of NETRI is to screen a lot of different compounds together and place that into what we call a digital library for each therapeutic indication. And the idea is when you have new drugs that you want to target, and you want to try and to compare the digital signature upon all the different compounds that are into our digital library, to see if this compound will have success or a failure to go into clinical trials when the compounds that you're screening into your digital library are properly qualified. So for that, we have developed a platform, and you can predict safety, efficacy, or diagnose. And for that, we have developed a platform in which we use compartmentalization devices. So in 2D or in 3D, here I'm going to go to example onto 2D only, in which you have on one compartment a cell body only, and on the other compartments only sprouting. And those sprouting can be done, and the compartmentalization is done by small microchannels that are 3 micron high. So as you can see, and the same idea as Mimetas, we are only using 96 well plate alignment reservoirs, in which per chips you have 16 different type of independent conditions. So basically, you seed cells body on one side, they sprout for approximately 3 weeks, and they are mature enough. You apply drugs and you record electrical activity in the cell bodies, but also in the axons themselves and in the sprouting. We have several type of architectures that are able to address different type of indication, and we are going to full 3D reconstruction system that are fully innovated and vascularized using our duplex link camera system. We integrated this whole package with a software application, and we've partnered with Axon Biosystem to record the electrical activity for an acute or chronic exposure of a compound. So there's a bunch of software behind that makes sense and plug into our digital library system. Now, that being said, we need the sensor itself, and this is the whole point of the cell interaction that I'm willing to present here. Again, neurons, especially sensory neurons, are very sensitive neurons in terms of HRPAC, and we need to qualify which targeted receptors we have at the endings of the synaptic buttons that will allow to make the sensing effect. So for that, we need to assess repeatability. We have different cell sources. So here I introduce, for example, axons, HIPSC derived cells. And before using in any screening compound with our customers, we always screen the batch-to-batch variability. Especially for neurons, we make them for each batch. We have a collections of quality control tests in which we test both immunofluorescence, as you can see, to look at the proper expression of markers, but also the electrophysiological activity. And we have some classical compound that we are testing within the batch themselves to assess if the receptors is present or not, and if the receptors have the proper interaction. So those are axons. Cells. We also have Fujifilm sensory cells, and you can see that we characterize them in the same way. So we have imagined, and now it's in production, a very quality control standard test to select different type of batches. This is, for example, for our CNS application with glutamatergic and GABAergic neurons, not specific to sensory neurons, but we have made this format that is available on our website. And bottom line, the quality control gives us the right batch to retain when upon specific application. So, for example, we have some customers that ask us to have certain type of sensors at the end of the projection. We qualify them every time, and in certain time of quality control, we have some failure, and this is where the interaction with the cell provider is very important to be in tight connection with our scientific team, because if there is something that is not expressed correctly and we want high throughput assays, this will bias completely the results of the assay. So we need to make this quality control systematically, and I cannot emphasize all the MPS provider to try to make the quality control before sending some results to the customers. So let me give you some couple of examples. Again, how we measure the electrical activity and how we can predict the efficacy of a compound. We always look at the differences between electrical activity states. So we don't claim that we have a perfect cell culture of perfect neuronal culture that is perfectly physiologically relevant, but we say that we have everything into the HIPSC culture, that when it's altered, we have the clinical effect of this alteration. So we measure the differences between the states of the reference state and the altered state, compound or reference compound. Now, let me look a little deeper into the sensitivity of this new readout. We have two ways of application of the compound, either what we call slow acting. So we look at the chronicle effect of a compound just with the same neurons and the same potential co-culture on the other side, and we apply the compound at T0, 1 hour, 7 days, up to 1 month in the chronicle aspects, and we record the electrical activity of neurons over time. And we look at the altered results in the percentage of change in the mean firing rate or the metrics that is interesting in the use of context. And we look at the difference of percentage of change. On the opposite, we can look with this method with an acute way to respond to a compound. So we've developed, it's the same idea, but not the same methodology. And in this particular case, we place the vehicle before and after positive control. So in any case, you can look at if the cell in the cell culture on the functional level is able to respond to a compound with the quality control that we expected with all the different assays that we're making in the same experiments. So this is very important to qualify the vehicle and the positive control response that is systematically the same. Because again, cell culture may be different, especially on functional level. In this particular case, we look at the expression of the difference of fold changing between the vehicle, the compound, and the positive control. So that's for the methodology. A couple of examples that we have developed with our customers. Temperature control, we were able to decode how specific neurons can have encode the difference of temperature, and this is relevant in terms of vaccine development, for example, or pain management into oncology. Proponet and LPS, you can see the difference of significant difference of change, and for pain management with lidocaine, where you can see that there's a significant change in the vehicle and lidocaine, and also there's a remnant effect over time. Another example with the fast-acting method, which is instantly recorded, paclitaxel and for pain and oncology, capsaicin, propionate, and ATP, and you can see systematically, you look at the vehicle, the KCL, and you can see that in both cases, we can compare with a statistically relevant manner the entire different type of chips that are applied in this at the same moment, and we always qualify that we have a positive and negative control over the vehicle and the targeted compounds. Now, if we look a little bit precisely into the specificity, and this is where this is an opening, the thing is that how we can exactly qualify and be objective in the way when you have a compound, the results, and the different, for example, with one only readout with the mean firing rate of neurons, will that be objective in the sense that you apply a compound and you have the response? Would you know if you have a response? Would you know which compound is it? So that's why we need more discriminating metrics not only for one or two metrics. So the way we do that is that with the Axon system and with globally general cell culture, you're able to extract up to 42 different network level metrics of neurons. And this is where the neural encoding is quite fine-tuning compared to the classical way to look at if you have a marker that is expressed in the cell culture, or you look at the metabolite of cell culture. Everything is designed by nature in which the neurons has the capability to grasp and to integrate different type of receptor into one different signal that needs to be after processed by the CNS. So when you look at all the different metrics, you have a 42 plane variables in which you can make a specific response of neural networks response of a compound. So we record everything into a 10-minute recording when we apply the compound in a fast or slow acting. And we just look at the changes, the changes of magnitude between the control and the altered state. We look at in all the electrodes that are spanned all over the chips, and we look at through the entire channel 1 to channel 3. And basically, we integrate that into what we call a digital signature of a compound. So everything is recorded here. We make the difference between before and after application, and we look it's relatively to reference states. Once we have this, we have what we call the digital signature, and we're able to look at all the different metrics that are discriminating between the experiment themselves. So we apply threshold, and we have a significant manner to not only have two metrics that can identify specific target compound of a mode of action of a toxicology, but to have more metrics that give us a more global and a plane of placing the different points together. Let me show you an example. For example, we have recently published a paper in which we can identify using this specific method into two different variables that are the best ones that are discriminated. Patients that have Alzheimer's and patients that are healthy. We have used the same method that I showed you before with specific neurons, and we apply cerebrospinal fluid of patients with AD and not with AD, and you can look that after a certain amount of application of CSF, we can really discriminate patients from one population to the control one. So again, this is only N equal 10 patients. It's a very preclinical study, but that shows that the potential of the method while using the best different metrics that can actually discriminate several populations. That was a global example. Again, we have a lot of several use cases in neuroinflammation, in neuromuscular junction, in gut-brain axis, cardiac, and a lot of different type of models that were created using neurons as a sensor. In 3D also, we are going into innovated organoids to have to record the electrical activity, what's happening into the organoids remotely without the necessity of complex electrode system. So I hope you have the concept, and feel free to contact us if you want any more information, or you have any new cell providers that have even more relevant receptors on the neurons. Thank you very much for your attention, and happy to answer the questions.
Thank you so much, Thibaut. That was very, very interesting. You have one question so far in the Q&A, so I'll just read it out for you. Well, there's a few questions. So first, how sensitive are these axons/receptor? Does this receptor density vary? And then what concentration of the stimuli do you have to introduce, and is it within physiological concentrations?
So indeed, there's a very important question to resolve of the concentration of receptors, and there's a huge variability between the cell providers, but also the maturity of the cell culture, and eventually the co-culture you place in the cell body. So we've measured everything by making different type of concentration of compounds that we know, and there's a variability that we need to characterize before. But this we cannot control because it's the cell providers that will make the number of receptors per electrode that we can have. But nonetheless, most of the receptors are expressed in a sufficient way to be as sensitive as placing subclinical concentrations of compounds that can be detected. So we can detect not only a very, very small concentration that is under what is classically delivered into clinical trials. There's not a lot of saturation on the opposite side. And we can also detect metabolic.
Thibault, I think you're frozen. I'm not sure if it's just on my end.
That may be generated by sorry, I got jet lagged or whatever. Can you hear me well?
That's okay. Yeah, I can hear you now. I wasn't sure if it was just on my end or not.
There was something going on, so I don't know how much of the response to get.
Yeah, we heard a good amount. Okay, yeah, Megan is verifying. She can hear you now as well.
Okay, I believe everything.
Yeah, so maybe just repeat the last few sentences that you were explaining.
So it's just about every concentration and density of receptors is dependent by from provider to providers, but most of the receptors are here, and we qualify the number of receptors by applying a known concentration that have a significant effect. Now, that being said, about the clinical concentration, we know that we can measure subclinical concentration, sub-concentration that are applied in clinical trials. So it's very, very thin in the way to measure the amount, and we can make dose dependency also at a certain level after you saturate the receptors. But since it's a dynamic system, you can wash and you can look at the dynamic also of the receptors being saturated.
Awesome. Thank you so much, Thibault. That's all we have time for questions right now.
Feel free to contact me anytime. Thank you very much.
Great. Thank you so much.
Hello, everyone. Good morning and good afternoon. I am Elisabetta Palama, Senior Research Scientist in React for Life. We are an Italian company, and we are based in Genoa, a city in the north of Italy, and we are an organ-on-chip provider. Our technology is called MIVO, which stands for multi-in vitro organ. It is a patented technology which comes from more than 20 years of research. It was also awarded in 2021 by European Commission and more recently by CPHI. It is an advanced in vitro platform which is able to maintain, to culture human tissue in a 3D controlled environment with a fluidic flow in the basal side which mimic the systemic circulation. These slides summarize the main features of our technology. First of all, the technology enables for the culture of 3D human tissues and also of patient biopsies. This allows also to have personalized therapies investigation. So the technology is not only compatible with 2D standard culture, but also to 3D organoids, for example, spheroids and all the 3D cultures. The drug can be administered directly in the circulation to mimic the systemic administration. And in this way, the drug is delivered by the physiological flow, which is in the physiological velocity, enabling and allowing to have more predictive drug testing. Moreover, the technology also allows to connect different chambers with different organs inside under the same fluidic environment to better study also the drug distribution and to study the crosstalk between different organs. This allows to carry out different applications, including disease modeling, but also pharmacokinetic studies. The MIVO technology is very versatile, and this flexibility is mirrored also by our different configuration. We have our single flow configuration, which is the one that I showed in the previous slide with the basal flow under the cultured tissue. And we also have our double flow configuration, which also has a second flow in the apical side and these allow to host interface tissues like, for example, lung tissue or intestinal tissue. Mostly we did all our validation in cancer field, and these allow us to develop our Mivo cancer on chip with different 3D culture models and different papers already accepted and validated. And also our Mivo cancer on chip allows to culture not only monolayer of cancer cells, but also matrix-based tumor models, spheroids, organoids, and even patient-derived biopsies. The system can also be complicated at different levels because we can, for example, include the drug administration in circulation to mimic the systemic administration. We can inoculate the immune cells to study the crosstalk with the tumor model, and we can either study the extravasation of immune cells to reach the tumor model, or also the invasion of the tumor cells. We carried out all our validation by creating 3D human cancer models based on the use of our alginate-based material, which allow a tunable stiffness, can be easily functionalized, it is optically transparent, and also has a very long-term stability. Indeed, here on the right of the slide, you can see a picture on confocal microscopy after 4 months of culture of cancer cells cultured in our alginate-based matrix. This allows also to carry out a different type of assays. I will show you very briefly some example. We were able to perform a drug efficacy assay by culturing a 3D human cancer model in our Mivo platform. First of all, we compare our model to the static model, and with attention to inoculate the chemotherapy drug, which was cisplatin, in circulation in the dynamic flow to mimic the systemic administration. And of course, in the untreated tumor model, all the cells were proliferating. Where we treated our model in static condition, we saw that dead cells were only present in the peripheral part of the 3D model, while when we treated our 3D cancer models in under dynamic, fluid dynamic condition, we noticed that dead cells were more homogeneously distributed in all the 3D model, also in the core. So this is due mostly to a more efficient penetration of the drug when we have dynamic condition, and is also allowed to carry out a more reliable drug efficacy test. Then we also compare our data with the in vivo model, and we noticed that we can have same regression in vivo and in vivo platform. Indeed, we observed comparable results, while static model led to unpredicted results. And moreover, we also were able to obtain these results in a faster way compared to the animal model, since in the animal model we need to wait, and we need to inject the tumor cells to wait for the tumor to grow and then to treat the animal. Then we can also, as I said in previous slides, the system can work in a multi-organ approach, in a multi-organ configuration. And in this configuration, different tissue models can be simultaneously cultured in different Mivo chambers and fluidically connected, like in vivo. And this configuration allows to carry out cancer metastasis assay, for example, to test how cancer cells detach from the primary tumor to reach a metastatic niche, how they become CTC, and which is the mechanism on the base of this process. And also, it's possible to run, for example, pharmacokinetic studies. We used this configuration in a recent paper by connecting fluidically a tumor model to a liver model with the cisplatin in circulation with the chemotherapy drug directly in the 3D flow. to study in the same experiment the efficacy on the tumor model and the off-target toxicity on the liver. Being aware of the importance of the immunotherapy, which are extremely growing in recent years, we also developed our immune chip by inoculating immune cells directly in the circulation, like it happens in the physiological condition in vivo. This allows to recapitulate the effect of the flow on the immune cell infiltration and also on their activation. We were able to co-culture immune cells and 3D cancer model in our MIVO platform, and we were also able to observe how the immune cells were able to extravasate to reach the 3D tumor. More specifically, we co-culture a neuroblastoma 3D tumor model with NK cells in circulation, and we observed that these immune cells were able to extravasate, so to lead the flow to reach the tumor model, the tumor chamber, and this extravasation was also tumor specific because it was significantly higher compared to a positive control, where the positive control is carried out by using the same matrix without tumor cells and with medium supplemented with 30% of MPS. Then we were also able to observe an infiltration of the NK cells inside the 3D model. Indeed, NK cells were able to infiltrate in the matrix and to interact with the cancer cells, and finally to induce the apoptosis. And which is very interesting and important in my opinion, we also observed that these data were completely in line with the clinical data, because the percentage of NK cells migrated from the flow was in line with the clinical reported data. This paved the way to personalized therapies, because we might think to use patient-derived material with patient-derived biopsies and also the immune cells from the same patient, and we might be able to predict the response to immunotherapy or chemotherapy for that specific patient. To summarize, my final slide, just to conclude with all the features of our technology. First of all, as I introduced at the beginning, the technology is compatible with the 3D cancer models and also with patient biopsies, which increase the predictivity of the study. It is very easy to sample the medium both from apical and basal side, enabling streamlined analysis. We are able to inoculate immune cells directly in circulation, like it should happen in our body in physiological condition, to study the physiological crosstalk between immune cells and cancer models. The platform is also PDMS free, which allows to carry out more reliable drug efficacy testing. Since the platform is not sealed, it's also possible to recover the sample at the end of the culture to perform multiple outcomes, for example, also omics, histology, or other type of analysis. And finally, again, the multi-organ connection allow to conduct also predictive pharmacokinetics or pharmacodynamic studies. Thank you. These are our contacts. Feel free to contact us if you have any more questions.
Thank you so much, Elisabetta. There are a few questions in the Q&A, which we have a few minutes for. The first is, what are the size of these tumor samples, and do they grow during the 120-day period that you mentioned?
They can go more than 120 days. We just reached the 4 months period. They were growing because maybe I didn't mention in the slide, but the staining was also for Ki67, which is a proliferative marker, and we were able to observe proliferation also after 4 months. Indeed, the dynamic flow allowed to have more efficient mass transport. And regarding the sites, consider that the size of the chamber is close the chamber is compatible with the 24 well insert, so anything can fit inside a 24 well insert can also fit in our chamber.
Great. And then they also ask, is there any central necrosis and how are these samples oxygenated?
We didn't observe necrosis inside, since, as I said, there is a more efficient and improved mass transport, so the growth factors can reach also the core of the model. And the second part of the question
was how the samples are oxygenated.
Through the medium. And so we just put it inside the incubator. With all the system goes inside the incubator, and the oxygenation is through the medium. And also the chamber is not sealed, so it's atmosphere pressure, so it can be oxygenated also through the lid.
Great. Thank you so much. And thanks again for your presentation. So that includes our talks for today. So I'll invite all of our speakers back online. Feel free to turn on your cameras, and we will do our roundtable Q&A. I will turn it over to Clive to primarily moderate. So if you have questions for a specific speaker or for all of the speakers or companies, please put that in the Q&A now and specify who your question is for. And I'll do my best to kind of moderate things. I don't know if we have any in the chat yet, so maybe we'll give people a few minutes. Maybe I could start with a kind of a brief question for each of the speakers. What would you say is the biggest challenge for your company at the moment with sourcing cells? I know, kind of a simple question, but yeah, Rachel.
Yeah, I think probably as I have mentioned in the slides, I think because we are reliant on our suppliers to obtain good quality, well, cells that have primary cells that have been isolated to high standards. It's that obviously with primary human cells, there is a limited supply. So that's why we have to continuously try to validate different donors. And I think that is the main challenge, that we're not just the only ones who are trying to use these primary human cells, because they are showing the specific phenotypes that we are looking for. And I think that's one of the, I guess you could say, a limitation of the primary human cells, that there is a non-continuous supply. Yeah.
Yeah, if anyone else wants to jump in and add anything, feel free. Okay, it does look like we have a question in the Q&A, and I think they don't specify, so maybe this is for everyone. Great talks. How do you manage to access patient data from the primary cells that are used? Example, drug sensitivity profile, and what are general criteria of each choice? Example, not above a specific age. So maybe we can just see if there's anyone off the top of their head that wants to start, or I can kind of just go through each speaker.
I think generally those questions will all depend on the ethics approval that go with the original tissue collection. So some ethics allow pretty minimal information. Other ethics do allow more, but you've got to justify why you're taking that additional information. So you're usually limited to age, sex, and any drugs that were used during surgery. But you can gain more information on that. I wonder if anybody can add to that, of course. Rachel.
Yeah, so I guess this is because this is something that we do work with closely with our suppliers. But yes, so there is the Human Tissue Act for us that does protect as well on the patient confidentiality and how we actually use the tissues. But yeah, so we do get, as you have said, several criteria to choose from, and that includes things like age, and some of them the cells would have been tested for certain conditions. There is also like a genotype that we can look at if there are specific mutations that our client have requests for example, in the NASH conditions. So yeah, and then we would then as a principle, we do try to select for primary cells in a wide range of criteria. For example, whether it's different ethnicities, different BMI, and things like that. We do take that into consideration so that we can also explore and validate a greater variety, and we are not biased in our selection of these cells that we validate. So yeah, there is criteria to choose from, but I think it's important to be able to also spread out that criteria and try to test as many as possible.
Is there anybody who would like to add anything to that? Then we do have another question. Actually, another couple of questions have come in. The next one is, how do we know that at this non-physiological PMS, I'm sorry, I don't know what that PMS is, the interstitial flow, shear stress, and adjusted blood oxygenation that happens at the level of tissues in human body, how can we be sure that these results we get are accurate? So probably a question, right? Yes, Elizabeth. Elizabeth, you flashed up and then flashed down. Were you going to answer?
Yes. So, first of all, regarding the shear stress and oxygenation, the only way is to perform computational simulation, which is always possible. And in my opinion, every study should be always carried out with also the computational simulation. And also, for the second part of the question, how can we be sure that the results are accurate? By comparing with clinical data, with animal data, to understand if our in vitro in vivo validation is right or not.
There's a few nodding heads there, so that's a positive. Another question's come in. Please address the following: number of cells, cell density, thickness of the tissue that are vastly different from true human tissue. Also, please discuss the dilutional effects of removing and refreshing the media and lack of monitoring proper oxygen. Thank you all. There's a couple of little questions in there. Does anyone like to try answering those? Can you see the questions that are up there, panel? Yeah. I think the first bit is quite easy, because the cell and cell density and thickness of tissues does depend on the context of use. So you can't sit and define something and then necessarily move that across to another completely different context of use. So I think that bit's quite an easy answer. So each of your different systems will require cells and cell density, etc., based on that use, and that will change even if it's going to be a small change in the context of use. So the dilutional effects of removing refreshing media and lack of monitoring proper oxygenation, I'll have to leave that to another person to answer. Rachel.
I think I can try to answer this. Yeah, of course, like the number of cells and the cell density, it is a lot of, I guess the questions posed here is a lot of it is worked out through development. Yeah, research and development. Quite a lot of it is through, trying to predict what the outcome will be, and then running the experiment itself, and then see what is the best, most optimized condition. So, like with regards to the number of cells and the cell density, that is usually worked out. Like it depends on the surface area, the volume of media, and all that, the size of your scaffold. And with regards to the thickness of tissues, it is, I mean, it is quite different in terms of even the MPS system itself. We are trying to simulate and mimic the human body, but ultimately, like for example, the shape of the organ or the organoid is also vastly different. But that's why you carry out further analysis to be able to understand how vastly different or similar it is to human conditions. As Elisabetta has said about comparing in vitro and in vivo studies to see how similar is the MPS system itself in trying to simulate an in vivo condition. With regards to the dilutional effects, I think first of all, it's I think that with regards to that, it may be more of a it can be a shock to the tissue when you are replenishing new media, and if it is particularly reliant on, like say, exchange of factors between multiple organs, like a dual organ system, for example, and that may be the case, but again, you have to carry out the proper assays and analysis to see what kind of stress that does result, or if it results in any functional differences. And with regards to the monitoring of proper oxygenation, there are bioreactors that provides a very controlled environment for that purpose. But from what we have seen, similarly to when you are culturing a plate of cells in an incubator, that condition is sufficient for our PMS system, for example. I hope that answers the questions.
Clive,
So
I think
would anyone like to add to that, or are you are we coming to an end? There's no new questions that have come up. I hope that the questions have been answered satisfactorily. Going back, just checking with the people who've asked the questions. I hope that the questions have been answered satisfactorily. Lauren, do you think we can wrap this up?
Yeah, I think so. Thank you so much, Clive, for your help.
Oh,
and thank you. Oh, yeah, I think just a thank you. Perfect.
Yeah.
So we got confirmation our questions were answered. So thank you again to all of our speakers for today and for all the interesting presentations. Just a reminder to everyone attending, the talks have been recorded, so we will be hoping to get those out to all the registrants as soon as possible. So thank you all for attending and for another great webinar, and we hope to see you at the next one. Thanks so much, everyone.
Thank you all. Bye.
Thank you.
Thank
Bye.
you.